Hi, today we'll be talking about biochemistry of cell. I don't know how to make this font bigger. If you know, please tell me in the comments. So if you feel that I'm speaking too slow, feel free to increase the playback speed at your end. We'll be we'll be starting with a case. A 20-year-old infant was presented with seizure, low muscle tone, decreased spontaneous movements, poor cry, refusal to feed. On examination, the baby had facial dysmorphism, high forehead, broad nasal bridge. On examination, the baby had facial dysmorphism, high forehead, broad nasal bridge. Lab tests revealed increased levels of very long chain fatty acids and increased levels of cholestanoic acid, which is a precursor of bile acid. And lab tests also revealed reduced plasmalogen levels in plasma. So, what is the probable diagnosis? Explain the biochemical profile and what is the current treatment strategy for this disorder. Diagnosis is most likely paroxysmal biogenesis disorder. It is a group of autosomal recessive disorders, also called as Zellweger spectrum disorders. Previously they were classified separately, but now they have been all put under Zellweger spectrum of disorders, in which the function of peroxisomes is partially or fully compromised. Zellweger syndrome, also called as cerebrohepatorenal syndrome, is the most severe form of peroxisome biogenesis disorder. It is due to defect in any of the 13 PEX genes, most commonly due to defect in PEX1 gene. PEX genes are responsible for assembly of peroxisomes. If there is a defect in those genes, peroxisomes won't be efficiently assembled, and hence the function of peroxisome will be compromised. In Zellweger syndrome, all functions of peroxisome are compromised. Very long chain fatty acid oxidation is compromised. Bile acid synthesis is compromised. Branch chain fatty acid oxidation, which is initially alpha oxidation, that is compromised. And synthesis of ether lipid plasmalogen is compromised. So, very long chain fatty acids and branch chain fatty acids, precursor of bile acids, accumulate in tissues, cause signs and symptoms of toxicity, and also their levels in plasma increase. Plasmalogen is required for myelin sheath formation, and because plasmalogen is not getting synthesized as a result of compromised function of peroxisome, myelination is defective, and that leads to seizures. Currently, there is no definitive treatment for this disorder. Prognosis is poor, and child usually dies in the first year of life. Current treatment only focuses on palliative care and improving the quality of life of the child for as long as it survives. And advancement in technology, we can only hope that there will be some definitive treatment in the future. Infantile deaf sum disease and X-linked adrenoleukodystrophy are other diseases in the Zellweger spectrum of disorders. We'll be studying about peroxisomes later. This was just to sensitize you to a clinical case. Is also asked as a MCQ in MBBS exam, or can be asked as a case in the exam. So coming to cells, a cell is the smallest structural and functional unit of an organism. As an independent unit, a cell can metabolize its own nutrients, provide for its own energy, it can replicate itself to produce succeeding generations of cells. Cells of all organisms have a nucleus, or they have a nucleoid which contains the DNA. Either they will have a nucleus or they will have DNA in the cytoplasm, and they have a cytoplasm surrounded by plasma membrane. Eukaryotes are membrane bound, they have membrane bound organelles suspended in the cytoplasm. Before going to that, we can see here in this diagram that the cytoplasm, plasma membrane, ribosomes, and genetic material which is nucleus in eukaryotes or nucleoid in prokaryotes is common to all organisms. Eukaryotes only have nuclear membrane and membrane bound organelles, and they are generally much larger in size compared to prokaryotes. You can see here the size difference. Cytoplasm is aqueous solution, that is the cytosol, plus suspended particles which can be ribosomes or the membrane bound organelles. When cytoplasm is centrifuged at 1,50,000 times G, G is the gravitational constant, what remains as supernatant is called cytosol. So this is actually called as ultracentrifugation. We'll be seeing later. On ultracentrifugation of cytoplasm, where G is the gravitational force of Earth, sediment contains membrane bound organelles and particulate matter, ribosome and proteasome. Ribosomes are protein synthetic machinery, proteasomes are protein degradation machinery of the cell. And the supernatant or the cytosol, it contains enzymes, coenzymes, inorganic ions, RNA, amino acids and nucleotides, and small organic molecules which are intermediates of various metabolic pathways. What limits the dimensions of the cell? Lower limit is probably set by minimum number of each type of biomolecule required by the cell. Smallest cells are bacteria mycoplasma, which are 0.3 micrometer in diameter, and cell volume is 10 raised to minus 14 ml. Upper limit of cell size is probably set by the rate of transport of nutrients into the cell and the waste products out of the cell. Balance between the two decides the maximum size the cell can attain. For a spherical cell, surface area is a function of square of radius, quite obvious, and volume is a function of cube of radius. Now, it is said that all organisms belong to three distinct domains of life. They have all come from a last universal common ancestor. This is the phylogenetic tree or family tree of living organisms. Last universal common ancestor became bacteria and archaea, and from the archaea there is another branch for eukarya. Bacteria and archaea are unicellular prokaryotes, whereas eukarya can be unicellular or multicellular, and they are obviously eukaryotes. Prokaryote, in Greek, pro means before, that means before nucleus, and eukaryotes, eu means good or true in Greek, Karyon in Greek means a kernel or nut. It's used for nucleus because cell's nucleus resembles a nut or seed in the cytoplasm. Bacteria can be seen in soil, surface waters, tissues of other living or decaying organisms. Whereas archaea, which have been recognized as a distinct domain by microbiologist Carl Woese in the 1980s, they are usually found in extreme environments like salt lakes, hot springs, highly acidic wetlands, in the deep depths of ocean. All eukaryotic organisms have evolved from the same branch that gave rise to archaea, and hence eukaryotes are said to be more closely associated to archaea than they are to bacteria. This is the difference between prokaryotic and eukaryotic cells. You can pause and read at your leisure. This is a diagram of eukaryotic cell, which is showing various organelles. Like eukaryotic cell, we have always been seeing a two-dimensional image, right? So this is a 3D image, beautifully showing the organelles, and this below here is also a section of a eukaryotic cell. And these are all electron micrographs, electron microscopic images of the various organelles like nuclear membrane, nucleolus, chromatin. This is the nuclear membrane, this is the nucleolus, this is chromatin, then rough endoplasmic reticulum, smooth endoplasmic reticulum, lysosome, mitochondrion, Golgi apparatus, this one. So this is another beautiful diagram which is showing plant cell here and animal cell. This is the animal cell and this is the plant cell. So plant cells have a vacuole which animal cells don't have. Plant cells have a cell wall which animal cells don't have. Plant cells have chloroplasts which we don't have. And this diagram beautifully will tell you the functions of all organelles. You can read at your leisure later as a revision. So finally we come to cells of humans or vertebrates. Similar cells make up tissue, tissues make up organs, organs make up organ systems, and organ systems make up an organism. Structures or organelles inside the cell can be enumerated as nucleus, endoplasmic reticulum, which is rough and smooth, Golgi apparatus, lysosomes, mitochondria, peroxisomes, ribosomes, cytoskeleton, cytoplasm, and the outer covering, cell membrane. Nucleus is the largest cellular organelle in humans. It is the carrier of genetic information in the form of DNA. DNA is bound to histones to form chromatin. Somatic cells have 46 chromosomes. Nucleus coordinates various activities of the cell. It's like the brain of the cell. Nucleus is separated from rest of the cell by an envelope that it's a double membrane organelle. It consists of two membranes and they are joined at nuclear pore, which are about 19 nanometer diameter. And the nuclear pore allows for bidirectional movement of molecules between nucleus and cytoplasm. The outer nuclear membrane is continuous with the rough endoplasmic reticulum. Some hormones interact at specific sites on DNA to regulate transcription. They are the steroid hormones, which have nuclear receptor, right? Some cells, like human RBCs, they lose their nuclei during maturation. This RBCs not having nucleus is an adaptation. RBCs also, human RBCs do not have mitochondrion. So the only source of energy for RBCs is glycolysis. And that is why RBCs do not use any oxygen which they are carrying, and this allows them to efficiently deliver oxygen to tissues. There is a dense non-membranous mass in the nucleus called nucleolus. It is closely associated with inner nuclear membrane. Nucleolus is the site for transcription. Nucleolus is also the site for assembly of ribosomal RNA. This is where ribosomal RNA is assembled and goes out into the cytoplasm at the nucleolus. Transport through nuclear pore is bidirectional. That is, the mRNA, which is formed as a result of transcription, it travels out of the nuclear pore for translation. Ribosomes, which are also synthesized in the nucleus, they need to move out into the cytoplasm. Their ribosomes are formed in the nucleolus, right? They need to move out into the cytoplasm so they can carry out protein synthesis. And proteins which are required for DNA replication, transcription and other processes in the nucleus, they travel to nucleus from the cytoplasm. These proteins have a specific sequence of amino acids known as nuclear localization signal. This signal directs them to the nucleus post-translation. When we study translation, we will know that translation has various steps, right from initiation to elongation, termination, and then there is post-translational modification and folding, and then it is that finally folded protein is direction, whether it will remain in the cell or whether it will go outside the cell. If it remains in the cell, whether it will go to the nucleus or the mitochondrion or some other organelle, that is decided at the end. Hutchinson-Gilford progeria syndrome, HGPS. We have seen Indian people at least have seen the movie by Indian actor Amitabh Bachchan. It's a disorder. It's caused by sporadic autosomal dominant mutation in the LMNA gene. The LMNA gene normally produces lamin A, which is a structural protein responsible for integrity of nucleus. When there is a defect in this gene, it produces a toxic mutant form of lamin A called as progerin that will attach. On one side, lamin A is not getting produced, which is essential for integrity of the nucleus, and a mutant toxic variant is getting produced, which attaches to the cell's nuclear envelope and it causes the cell to become unstable. This leads to premature cell aging and prevents efficient cell division and cell repair. Progeria is a very rare disorder, 1 in 5 million live births. Although it is a autosomal dominant disorder, so even one chromosome out of the two being defective is enough. It's a sporadic mutation. What's a sporadic mutation? One that occurs spontaneously in early embryonic development or in the sperm. Sporadic mutations are rarely inherited because they mostly occur spontaneously in early embryonic development or they occur during spermatogenesis. Coming to the second organelle, the endoplasmic reticulum. We saw that the nuclear membrane, outer membrane of the nucleus, is continuous with the rough endoplasmic reticulum. These are network of membranous tubules in the cytoplasm. The rough endoplasmic reticulum is involved in synthesis of certain proteins, not all proteins. Ribosomes attached to rough endoplasmic reticulum gives them this rough appearance. Proteins produced on ribosomes of rough endoplasmic reticulum enter the lumen of rough endoplasmic reticulum and are transported to another membrane system, which is the Golgi apparatus, the Golgi complex, in vesicles. And then these proteins which are made in the rough endoplasmic reticulum can have one of the following three fates. Proteins synthesized in RER can either be secreted out of the cell or they can be sequestered into membrane bound organelles like lysosome. What is the meaning of sequester? Sequester generally means to isolate or separate or hide away. But sequester in science or biology refers to absorb or to store. So sequestered in lysosome means they are absorbed by the lysosome and stored in lysosome and further processed in lysosome. Or they can be embedded in the plasma membrane. Proteins encoded by the nucleus and found in cytoplasm, peroxisomes or mitochondria. So proteins which are not destined to go to lysosome, proteins which are not destined to be in the plasma membrane, or proteins which are not destined to be secreted out of the cell, proteins which are supposed to be in the cytoplasm or in the peroxisomes or in the mitochondria, they are synthesized on free ribosomes in the cytoplasm. Smooth endoplasmic reticulum does not have ribosomes. Function is lipid synthesis like triglycerides, phospholipids, synthesis of steroid hormones. Smooth endoplasmic reticulum also contains cytochrome P450 oxidative enzymes, which are involved in metabolism of drugs and toxic chemicals like ethanol, alcohol. Glycogen is stored in regions. Glycogen is stored in regions of liver cells which are rich in smooth endoplasmic reticulum. Glycogen is stored in cytoplasm as granules, which are typically deposited directly on the external surface of smooth endoplasmic reticulum. Smooth endoplasmic reticulum of the liver has glucose 6-phosphatase. Liver is the only organ which has glucose 6-phosphatase. That is why the glucose which is produced by glycogenolysis can be converted to free glucose into the bloodstream. And that is how liver maintains blood glucose levels. Whereas skeletal muscle does not have glucose 6-phosphatase, and therefore the G6P or the glucose 6-phosphate, which is formed as a result of glycogenolysis, has to be used by muscle for energy, and hence muscle cannot contribute to blood glucose. Please don't write G6P in your exams. Always write full form glucose 6-phosphate. What is the clinical relevance of smooth endoplasmic reticulum? Huge clinical relevance. The enzymes of smooth endoplasmic reticulum, as we saw here, they also have the cytochrome system for detoxification of compounds, foreign substances, and that is where it is linked to chronic alcohol ingestion, which increases the microsomal ethanol oxidizing system, also called as MEOS. And this microsomal ethanol oxidizing system converts ethanol, NADPH and oxygen to acetaldehyde, NADP and water. Now, breakdown by alcohol dehydrogenase. Alcohol dehydrogenase is the classical pathway for ethanol detoxification, which reduces ethanol to acetaldehyde, and it uses NAD as the coenzyme. NAD gets reduced to NADH plus H plus, which is actually a good thing, right? Then this NADH can go into electron transport chain and give rise to energy in the form of ATP. Or if pyruvate is converted to lactate, that NADH can get converted to NAD again. It need not go into electron transport chain. But when alcohol is consumed in excess for a long period of time, it leads to activation of microsomal ethanol oxidizing system, and the main enzyme is CYP2E1. CYP2E1 is the main enzyme of the microsomal ethanol oxidizing system. It does not use NAD. In fact, it is yes, I made a mistake just a while ago. Alcohol dehydrogenase is dehydrogenase. So it's really beautiful. Alcohol dehydrogenase indeed causes dehydrogenation or oxidation of ethanol to acetaldehyde, and NAD becomes NADH plus H plus, which is a good thing, right? But when alcohol is consumed in a large amount for a long period of time, microsomal ethanol oxidizing system is a mixed function oxidase. It uses cytochrome P450, right? It's a group of enzymes, and in that it uses CYP2E1. So it is indeed causing oxidation of ethanol, that is, hydrogen atoms are removed from this ethanol, right? But it is causing simultaneous reduction of O2. Cytochrome P450 or Cyte2E1 microsomal ethanol oxidizing system needs both O2 and NADPH to carry out oxidation of ethanol. So NADPH is being an electron donor to Cyte2E1 enzyme, and that enzyme will pass the electrons on to oxygen, molecular oxygen, and so NADPH is getting converted to NADP, and it is also getting hydrogen from ethanol, and oxygen is getting converted to water. So electrons from ethanol and NADPH go to oxygen and becomes water. Ideally, that is what is supposed to happen. But as this Cyte2E1 carries out this reaction, sometimes that conversion of O2 to water does not happen completely. There are a lot of reactive oxygen species, ROS, which are produced right from superoxide radical to hydrogen peroxide H2O2 to hydroxyl radical. So all these reactive oxygen species are produced because of incomplete conversion of oxygen to water, and that causes a lot of damage, as we will see. This chronic alcohol ingestion activates this system and causes increased tolerance to alcohol, because chronic alcohol is metabolized faster than a non-alcohol drinker, and that alcoholic needs the same amount of alcohol to get the high next time, as in the dose of alcohol required goes on increasing. There is tolerance. There's also tolerance to cross tolerance to medications like sedatives, anesthetics, anti-seizure drugs, which is a bad thing, because it is metabolizing this cytochrome P450 is also metabolizing these drugs, and they are getting cleared faster, and so their efficacy also goes down. And there is oxidative stress and cell damage. Why? I just now said that this conversion of ethanol to acetaldehyde, it doesn't happen, the oxygen doesn't completely get reduced to water. There is a lot of formation of reactive oxygen species from superoxide radical to hydrogen peroxide to hydroxyl radical, in between reactive oxygen species intermediates, and they cause oxidative stress and cell damage, lipid peroxidation, fatty liver. Fatty liver in a chronic alcoholism causes microsomal ethanol oxidizing system in overdrive. Increased toxicity of common chemicals like household industrial solvents like dry cleaning fluid, carbon tetrachloride, drugs like acetaminophen. Cyte2E1, which is primary driver of MEOS, converts these relatively harmless substances into toxic compounds. So on one hand there is cross tolerance to drugs, and there is increased toxicity on the other hand to some other drugs which are converted to toxic substances by overactive microsomal ethanol oxidizing system. So how does increase in MEOS cause oxidative stress? Firstly, as we discussed, that oxygen is not completely reduced to water. There is a lot of production of free radicals like superoxide radical, hydrogen peroxide, and hydroxyl radicals. So that they will cause damage, lipid peroxidation, that is the damage the membrane lipids and cellular integrity. And then antioxidants of the cell. NADPH is required to keep glutathione in reduced state. And when this glutathione is the one that neutralizes reactive oxygen species, right? Glutathione, which is in reduced state, neutralizes reactive oxygen species, but when it neutralizes reactive oxygen species, it itself gets oxidized, right? And that needs to be reduced again, and NADPH is the one to reduce it. And if this MEOS is consuming NADPH, there is no NADPH left to reduce oxidized glutathione back to reduced state, so it can neutralize free radicals. That is also another cause why there is damage to liver cells in chronic alcoholism. And these MEOS acetaldehyde which is formed, there is also hydroxyethyl radical which is formed in this reaction. They can directly bind to liver proteins and they can damage the liver proteins. And this is how MEOS causes oxidative damage to liver. The adjective microsomal is derived from experimental cell biology that is sometimes used for processes occurring in endoplasmic reticulum. There is no organelle as such as microsome in the cell. What happens is when the cells are lysed in lab, endoplasmic reticulum gets fragmented to vesicles called microsomes. These microsomes can be isolated by centrifugation. And that is why anything related to the endoplasmic reticulum is called as microsomal. Endoplasmic reticulum is the primary, smooth endoplasmic reticulum is the primary reservoir of intracellular calcium ion. So much so that calcium concentration in endoplasmic reticulum can be 10,000 times that of surrounding cytoplasm. By sequestering or isolating and then releasing calcium as and when required, endoplasmic reticulum, smooth endoplasmic reticulum regulates cellular homeostasis and it triggers vital physiological processes like muscle contraction and nerve signal transmission. How smooth endoplasmic reticulum regulates calcium? It isolates calcium. There are pumps continuously pulling calcium from cytoplasm into SCR. Then there are calcium binding proteins in the smooth endoplasmic reticulum like calreticulin, calsequestrin. They bind to calcium and store it in smooth endoplasmic reticulum. And then in response to cellular signals as and when required, smooth endoplasmic reticulum opens ion channels like IP3 receptors or ryanodine receptors and releases calcium into the cytoplasm. In muscle, smooth endoplasmic reticulum is also called as sarcoplasmic reticulum and it releases calcium to cause muscle contraction. Cytosolic calcium also acts as a second messenger for various signaling pathways. If the calcium levels of smooth endoplasmic reticulum drop due to some reason, they trigger something called as endoplasmic reticulum stress. They cause endoplasmic reticulum stress. Why is that? Because calcium is required for chaperone mediated folding of proteins in the endoplasmic reticulum. Proteins are formed in the rough endoplasmic reticulum and they fold in the smooth endoplasmic reticulum using chaperones. Chaperones like calnexin, calreticulin, they are calcium dependent chaperones. Calreticulin, chaperone protein. When calcium is decreased, proteins are not able to fold properly and there is accumulation of improperly folded proteins in the cell which causes endoplasmic reticulum stress, and response to this stress is unfolded protein response. which can lead to cell death if there is a large accumulation of such misfolded proteins. Hypocalcemia or low calcium levels in the blood can cause endoplasmic reticulum stress, but there are several mechanisms for hypocalcemia causing cell necrosis, and endoplasmic reticulum stress is just one of those. Coming to Golgi apparatus, Golgi is a complex involved in modifying proteins synthesized in rough endoplasmic reticulum, and then those proteins synthesized in rough endoplasmic reticulum are sorted and distributed, and they can be taken to the lysosomes or they can be transported out of the cells in secretory vesicles or they can be inculcated in the plasma membrane. Golgi apparatus consists of stack of flattened vesicles in the cytoplasm. These flattened stack pouches are called cisternae. Right? You can see these, and they are divided into three compartments: the cis Golgi network, medial Golgi stack, and trans Golgi network. Cis Golgi network is the one facing the nucleus. It's convex. Medial is in between all of these. And then the trans Golgi network is one which is facing the cytoplasm. One which is facing the plasma membrane. So the proteins or anything that is synthesized in the endoplasmic reticulum, it comes to the Golgi apparatus in the form of incoming transport vesicle, and then it is processed in the Golgi in the medial stack, and then whatever is processed, that can then from here the newly forming vesicle, the secretory vesicle, from the trans side it can go to lysosomes or it can go outside the cell or it can go into the plasma membrane. Golgi complex also participates in post-translational modification of proteins, like addition of complex branch oligosaccharides to the protein, sulfation of proteins, phosphorylation of proteins. Golgi complex plays a role in activation of cholera toxin. Cholera toxin is endocytosed in caveolae vesicles, which subsequently merge with lysosomes. The acidic environment in the lysosome contributes to activation of cholera toxin, and as the toxin is transported through the Golgi and endoplasmic reticulum, the toxin is further processed and activated. After Golgi, we come to lysosomes. They are intracellular organelles of digestion. They are also the waste disposal system of the cell. They are enclosed by a single membrane, and that is just to keep the enzymes in the lysosome compartmentalized. They are central to a wide variety of body functions which involve eliminating unwanted material and recycling the components, like destruction of infectious bacteria, yeast, recovery from injury, tissue remodeling, involution of tissues during embryonic development, and normal turnover of cells and organelles. Nucleases, phosphatases, esterases, glycosidases, proteases, they are all examples of lysosomal digestive enzymes. Most lysosomal digestive enzymes are hydrolases. That is, they break single bonds by addition of water. There is a enzyme group called cathepsins, right, which are cysteine proteases. They are a biomarker of lysosomes. Increase in plasma level of cathepsin, lysosomes are getting broken. This diagram gives us an amazing synopsis of reactions in the ribosomes. You can take a look at your leisure. The hydrolases have optimum pH of around 5.5. That means above that and below that pH, the enzyme will have lesser activity. This pH of 5.5 is maintained inside the lysosome by actively pumping protons into the lysosomes. And if at all these enzymes leak into the cytoplasm, they will get inactivated because cytoplasm has pH much higher than that, around 7.23. So it is protected from the hydrolases accidentally released from lysosome. And anyway, they are also bound by this membrane. Lysosomal storage disorders are genetic defects in lysosomal enzymes or in the proteins required for targeting enzymes to lysosomes. So, mannose 6 phosphate receptor is one such protein required for targeting enzymes which are synthesized elsewhere. So, lysosome does not synthesize its own proteins. Enzymes have to be taken to lysosome from somewhere else. And when there is a defect in these enzymes, it leads to accumulation of undigested material in lysosomes. The accumulation may be so extensive that normal cellular function is compromised. This happens particularly in neuronal cells. These disorders are called lysosomal storage disorders. There are two main examples. To note one thing, that most of the lysosomal storage disorders are autosomal recessive. So these two examples, both of these, Tay-Sachs disease and Pompe disease, are autosomal recessive. But one key difference between Tay-Sachs disease and Pompe disease is that Tay-Sachs is mainly neurodegenerative, that is, it affects mainly the neurons, whereas Pompe is neuromuscular, having a lot of effect mainly on the muscle cells. We'll see how. Tay-Sachs disease is neurodegenerative disorder and autosomal recessive genetic disorder which causes buildup of partially digested gangliosides in the lysosomes of brain and spinal cord. And that is why those cells then get damaged. Pompe disease neuromuscular, autosomal recessive due to deficiency of enzyme acid alpha glucosidase, which leads to accumulation of glycogen particles in the lysosomes of cells, leading to varying degrees of muscle weakness among other symptoms. Both these Tay-Sachs and Pompe disease are autosomal recessive, as are most of the lysosomal storage disorders. But Fabry disease and Hunter syndrome are lysosomal storage disorders which are X-linked. Coming to mitochondria. Mitochondria are oval-shaped organelles whose primary function is to generate large amounts of energy in the form of ATP. They are nicknamed powerhouse of the cell. Mammalian cell has 800 to 2500 mitochondria. Number per cell varies depending on the metabolic activity of the cell. RBCs don't have mitochondria because we don't want the RBCs to utilize the oxygen which they are carrying. Liver and muscle cells are rich in mitochondria. Mitochondria are unlike other cell organelles. Most cell organelles are single membrane bound, like lysosomes, peroxisomes, endoplasmic reticulum. But mitochondria have two cell membranes, and very importantly, mitochondria are unlike other cell organelles because they have a unique genome or mitochondrial DNA. The organelle has compartments that carry out specialized function: outer membrane, intermembrane space, inner membrane, cristae, mitochondrial matrix. Outer membrane is rich in lipids, phospholipids, cholesterol. Outer membrane is freely permeable to smaller molecules. It is said that outer membrane is freely permeable to it is said that outer membrane is freely permeable to molecules up to 1000 Dalton, and that is because it has these transmembrane proteins called porins which serve as free passage. Outer membrane also has very important enzymes for elongation of fatty acids, oxidation of epinephrine, degradation of tryptophan. Outer membrane can associate with endoplasmic reticulum in a structure called MAM, mitochondria associated endoplasmic reticulum membrane. This is not involved in transfer of lipids between endoplasmic reticulum and mitochondria. Sorry, this is involved. MAM is involved in transfer of lipids between endoplasmic reticulum and mitochondria. Inner mitochondrial membrane. This is a beautiful diagram you can see here of the mitochondrion. Inner mitochondrial membrane is rich in proteins. Outer mitochondrial membrane was rich in lipids. Inner mitochondrial membrane is rich in proteins with protein to lipid ratio 3:1. And inner mitochondrial membrane is not at all freely permeable. In fact, it is impermeable to most small molecules and ions, including protons. And we need specialized transport proteins to regulate passage of metabolites into and out of mitochondrial matrix. Inner mitochondrial membrane also has proteins of ETC, complexes 1, 2, 3, 4, ATP synthase, complex 5, and transport proteins. Mitochondria have the ability to divide, and if they lose this ability, it can cause diseases in the cell. Inner mitochondrial membrane has a phospholipid cardiolipin, which is a signature phospholipid of inner mitochondrial membrane, which means cardiolipin is almost exclusively found in inner mitochondrial membrane. It is a very important molecule. It stabilizes the proteins of ETC, helps in folding of inner mitochondrial membrane, and acts as signal for apoptosis in times of cellular stress or damage. Apoptosis is programmed cell death. That means if the cell feels that it is in danger or it has got damage beyond repair, it can kill itself before it is killed by someone else. Necrosis is forced cell death due to injury, but apoptosis is programmed cell death. Apoptosis can be physiological or apoptosis can be pathological. That means even normally when cells are have completed their lifespan, they undergo apoptosis, so that is physiological. but they can undergo under pathological conditions. Whereas necrosis is death of cells, which always happens under some insult. Necrosis is not a physiological process, is always pathological. Inner membrane of mitochondria is folded and invaginations of inner membrane are called cristae. Folding enhances surface area and thus the ability of mitochondria to produce energy is enhanced. See, more ETC complexes are packed in a lesser space, and that is how the ability of mitochondria is enhanced. Cells that have greater demand for ATP have more cristae. Quite obvious. You see, these are cristae. These, the invaginations of the inner mitochondrial membrane. Mitochondrial matrix is enclosed by inner mitochondrial membrane. Matrix has a mixture of enzymes, ribosomes, tRNA, mitochondrial DNA. It has enzymes for beta oxidation of fatty acids, ketone body synthesis, TCA cycle. It has pyruvate dehydrogenase complex. Pyruvate is transported into mitochondrial matrix along with the proton. This is an example of secondary active transport. We'll see in the next chapter, next lecture. Most of the enzymes and proteins present in mitochondria are coded for by the nuclear DNA and they are synthesized by free ribosomes in the cytoplasm. Mitochondrial DNA is circular and codes for about 13 proteins, which are an important part of ETC complexes. Complex 2 of ETC, complex 2 of ETC, succinate dehydrogenase complex, is coded for purely by genomic DNA. That is the DNA in the nucleus, nuclear DNA. So in mitochondrial disorders, complex 2 or succinate dehydrogenase complex is not affected. So one of the lab diagnoses of mitochondrial disorders is succinate dehydrogenase positive but cytochrome C negative. Cytochrome C is not found in mitochondrial diseases, but succinate dehydrogenase will be positive. Mutations in mitochondrial DNA lead to genetic disorders which have maternal inheritance. Mitochondrial important point in mitochondrial heredity is that it has maternal inheritance because mitochondrial DNA comes purely from the ovum. The DNA, the mitochondria of the zygote come from the ovum and sperm has no contribution. Examples of mitochondrial diseases are Leber hereditary optic neuropathy or myoclonic epilepsy with ragged red fibers, MERS syndrome. There are a lot of mitochondrial DNA disorders. They mainly affect the brain, heart, and there are a lot of mitochondrial DNA disorders. They mainly affect the brain, heart, and skeletal muscle. And they have incidence of about 1 in 5,000 live births. 1 in 200 healthy individuals may be carrier of pathogenic mitochondrial DNA mutation. And mitochondrial DNA diseases are important because They have such high incidence, 1 in 5,000 live births, which is more than all of the disorders of any of the cell organelles genetic diseases that we will see. Higher incidence. And that's why mitochondrial DNA disorder should always be considered as a possibility in multi-system disorders involving the brain, heart, and skeletal muscle with a maternal pattern of inheritance when there is no explanation for symptoms. Other functions of mitochondria: thermogenesis in brown adipose tissue of newborn, steroid hormone synthesis in steroidogenic tissues. Mitochondria also play a central role in apoptosis. Cytochrome C is released from inner mitochondrial membrane in irreversible cell injury that when it enters the cytosol activates caspase 9, which is a key mediator of apoptosis. Mitochondria are also direct targets of thyroid hormones and certain steroid hormones, which is how those hormones increase ATP production and metabolic rate. Mitochondria also act as second messenger in broader endocrine pathways by managing intracellular calcium and reactive oxygen species, which is the role of mitochondria in hormone signaling. Peroxisomes are similar in size to lysosomes. They are also single membrane bound. Peroxisomes are also single membrane bound like lysosomes. But their function, peroxisomes, is slightly different. Peroxisomes are involved in oxidative reactions using molecular oxygen. These reactions produce toxic. Any reaction which uses molecular oxygen, to some degree, they will produce reactive oxygen species, including hydrogen peroxide, which in peroxisomes is degraded by an enzyme called catalase and some other enzymes. So, catalase can be a marker enzyme for peroxisomes. Peroxisomes play a role in 1, 2, 3, 4. Beta oxidation of very long chain fatty acids. That is aliphatic side chain of 22 carbon or more, or that means overall carbon of about 23 or 24 carbons or more than that. Branch chain fatty acids initially undergo alpha oxidation and then undergo beta oxidation. So initial alpha oxidation of branch chain fatty acids occurs in peroxisomes. Conversion of cholesterol to bile acids, part of it happens in peroxisome in the sense that the bile acid precursors are synthesized in endoplasmic reticulum. They are 27 carbon molecules. They enter peroxisomes and they are converted by beta oxidation to 24 carbon cholic acid and quinodeoxycholic acid, the primary bile acids. And then these cholic and quinodeoxycholic acid are conjugated with taurine or glycine within peroxisome itself, which makes them more water soluble, less toxic. Golgi apparatus helps in transport of these bile acids into biliary canaliculi. Peroxisomes also synthesize ether lipids, plasmalogens. They play a part in synthesis of these lipids, we'll see. Now, why should peroxisomes oxidize fatty acids in the first place? Because mitochondria cannot oxidize very long chain fatty acid due to lack of transporters. Mitochondria doesn't have transporters which will transport these fatty acids inside the mitochondrial matrix. Therefore, they must first be oxidized in peroxisome and then long chain fatty acids, which are 14 to 20 carbon, they still need the carnitine shuttle for transport into mitochondria. Short and medium chain fatty acids. Medium chain are those which are generally less than 12 carbon. They can diffuse directly into mitochondrial matrix. Very long chain fatty acids are transported into peroxisome as esters with coenzyme A via peroxismal ATP binding cassette half transporters, primarily the ABCD1, also called as ALDP or adrenoleukodystrophy protein. Adrenoleukodystrophy protein ALDP. ABCD1 is ATP binding cassette subfamily D member 1. Defect in this gene coding for this protein causes X-linked adrenoleukodystrophy, which is a paroxysmal biogenesis disorder. Unlike standard phospholipids, which bind to fatty acid via ester bond, plasmalogens have a vinyl ether bond, which acts as a sacrificial target for free radicals. Plasmalogens absorb the damage, sparing other vital molecules. Peroxisomes only initiate the plasmalogen synthesis. Ether bond is formed in peroxisomes, but plasmalogen synthesis is completed in endoplasmic reticulum. You see here, this is a regular diacyl glycerol phospholipid. This is a ester bond phosphate group, right? Whereas this is plasmalogen. There is phosphate group, ester bond, and in SN1 position there is vinyl ether bond. Vinyl ether is carbon double bond carbon, and that carbon is bound to oxygen. So vinyl ether bond. This double bond, this gives greater flexibility to the structure. Ribosomes are nucleoprotein. That means they are a complex of rRNA and protein. Ribosomes bind mRNA, tRNA to synthesize polypeptides and proteins. Ribosomes, being entirely particulate, do not have a membrane. And they are often called non-membranous organelles. Ribosomes have two subunits. Eukaryotic ribosomes are ATS. Smaller subunit reads mRNA. Larger subunit catalyzes the formation of peptide bond between adjacent amino acids of the polypeptide chain. Smaller subunit is 40S, larger subunit is 60S, combined ribosome is 80S. S is Swedberg unit, measures particle sedimentation rate when spun in an ultracentrifuge and is an indicator of particle size and density. It depends on mass and shape. When two subunits of the ribosome, 60S and 40S, combine, resulting structure is more compact and it sediments differently. Therefore, 60 plus 40 is not equal to 80, and 60 plus 40 is 100, but in reality it has not become 100. The sedimentation coefficient, Swedberg unit for the ribosome is 60 plus 40, 80, even though it is not equal to 80. Ultracentrifugation is if you centrifuge at rates above 40 to 50,000 revolutions per minute, rotations per minute, and centrifugal force is more than 50,000 G. And this, because of the high force and high speed, we need vacuum and refrigeration to prevent friction and heating. You can see this is from protein data bank. There is 40S ribosome, 60S ribosome. This is eukaryotic, and this is prokaryotic, 30 plus 50, and combined is 70S prokaryotic. You can see that the eukaryotic ribosome is slightly more complex compared to prokaryotic ribosome. Ribosomes can be free or membrane bound depending on their spatial distribution in the cell. If they are attached to endoplasmic reticulum, they are bound. Otherwise, they are free in the cytoplasm. Beauty is that two subunits of ribosome only attach to each other at the time of translation. At the exact time of translation. When the cell is not synthesizing proteins, the two subunits are floating freely in the cytoplasm. That means even the ribosome subunits bind to the endoplasmic reticulum as well just before translation. When they are not translating, they are floating freely in the cytoplasm. Thus, ribosome binding to endoplasmic reticulum is a dynamic process. So this finishes the current lecture. We'll discuss the remaining cell organelles. We'll discuss the cytoskeleton and the cell membrane in the next lecture, because in the next lecture I'll be discussing the transport across cell membrane. So it is better to study the cell membrane structure in the next lecture, and also the cytoskeleton, because already this has become a very long video. Thank you for your patience and have a great day, and see you in the next video.